Welcome to the National Institute of Technology, Gunma College, situated approximately 100 kilometers northwest of Tokyo. Here, you'll find the Engineering Medical Nanotechnology Laboratory, overseen by Professor Alexandre Loukanov. Specializing in the design and development of nanoparticles, our focus extends to various applications, notably in the field of cancer research. Nanoparticles used for medical purposes can vary widely depending on the specific application. Some common types include liposomes are known as lipid-based nanoparticles used for drug delivery due to their ability to encapsulate drugs in target-specific cells or tissues. Polymeric nanoparticles. They are made from biocompatible polymers, such as poly-lactic-coglycolic acid, which are used for drug delivery, imaging, and tissue engineering. Metallic nanoparticles, which are made from metals like gold, silver, or iron, which have unique optical, magnetic, or catalytic properties used in diagnostics, imaging, drug delivery, and therapy. The vaccines gene therapies involve packaging of drug substances such as messenger RNA into nanoparticles that deliver the drug where it is needed in the cells or the body. RNA is formulated into lipid nanoparticles by mixing RNA and buffer with polymer and lipids to initiate self-assembly. As the fluids mix on first contact the cationic lipids complex with the negatively charged nucleic acids to form the beginning of the particle core. Finally, the lipids orient themselves on the surface with hydrophobic tails pointing inwards. In a similar way can be produced other nanoparticles as drug delivery systems with high drug loading and good stability. Every day, inside the body, tiny disease-fighting immune system cells called T cells find and destroy invaders like bacteria, viruses, and cancer cells. The drug delivery nanoparticles insert the messenger RNA into the patient's own T cells. This mRNA is a genetic instruction to the T cells to grow an artificial receptor called CAR, or chimeric antigen receptor, that can recognize and attack cancer cells in the body. Millions of copies of these genetically engineered CAR-T cells are grown in laboratory and injected back to the patient intravenously. CAR-T cells then circulate through the body. They bind and kill cancer cells and multiply, so even more T cells can join the fight. The CAR-T cells have been tested against cancer as leukemia and lymphoma. They target the antigen CD19, which is on the cancer cells. Thus, they trigger the cancer cell's death. As one cancer cell dies, the CAR-T cells move on to the next until all cancer cells are destroyed. Now we can see some cancer cells through a microscope. Cancer cells have changed in their DNA which make multiply out of control. These changes make cancer cells different from normal cells. Here we can see some green CAR-T cells through a microscope. Once they are in the body, the special receptors enable the CAR-T cells to seek out and recognize the patient's cancer cells. We are now looking at the magnified surface of a CAR-T cell in contact with the cancer cells. The CARs can directly bind to specific markers on the patient's cancer cells shown in yellow, overcoming the need for MHC1 to be present. Then a cluster is formed. We will now move below the cell membrane. Here we are looking inside the CAR T cell at the CAR signaling domains. The binding interaction has activated the signaling domains, beginning a chain of events which activates the CAR T cell. Once activated, the green CAR T cells release toxic chemicals into the cancer cells, which kills them, indicated in red. CAR T cells are known as serial killers because they can kill, release, and move on to kill other cancer cells in a cycle that can occur multiple times. This makes them very effective in fight against cancer. CAR T cells can remain in the body and will reactivate if the cancer returns even years later. Next, let's see some data of clinical trials showing dramatic and rapid regression of a deadly form of brain cancer through CAR T cell therapy. There was documented dramatic and rapid regression of glioblastoma. This type of treatment can be very helpful in treating some types of cancer, even when other treatments are no longer working. The nanotechnology is promising to revolutionize cancer therapy. The scientists are making great progress discovering new ways to improve the diagnosis and treatment of cancer. The nanoparticles are incredibly tiny, and they can be administered to patients intravenously. They easily float through the bloodstream, seeking out cancer cells wherever they may be hiding. Some nanotechnology is designed to seek out cancer cells and attach to them using specialized receptors which bind to the tumor selectively. Once bound to the tumor, the nanoparticles send out signals that tell doctors where it's located. This can help accurately visualize tumors that would not show up on conventional medical scans. Other nanotechnology is designed not only to find cancer cells but to kill them selectively using specific receptors that attach only to the tumor without injuring nearby normal tissues. With time, the nanoparticles are eliminated from the body. These are only two applications, but many other applications are developing in the near future. Thank you very much for your kind attention.